everyone, good day. Welcome back to my channel. So in today's topic, we'll be talking about bacterial control. So as you can observe to our background, you can see the different types of disinfectants, no? So in controlling microorganisms. So later on, we'll be able to identify the difference between disinfectants and sterilization. So, but before we proceed, we'll be able to discuss first the objective. So, ano ba yung ating malalaman dito sa ating topic na to? So, at the end of this lesson, the students must be able to list down different methods of controlling microorganisms. Number 2, describe the effects of each antimicrobial agent in controlling microbial growth. Number 3, discuss the importance of these methods in assuring sterility in healthcare facilities and healthcare practices. As a medical professional, we must be able to identify how to control microorganisms in order for us to contain and to control these microorganisms so that we cannot spread these microorganisms to other humans or to other people that we met. Okay, so let's start. So terms related to destructions of organisms. But before we proceed, let's know first the different terms. So control of microbial growth. So number 1 is sterilization. Killing of all microbial life. So meaning pinapatay ang lahat ng mga microbial life. So commercial sterilization so only has enough to kill Clostridium botulinum or canned food. So there are some commercial sterilizations, no, just like canning. Okay, yung nagtatrabaho sa canning, lahat ng mga canned goods natin just like the sardines, no, 555 sardines, family sardines or kahit anong mga sardines, gumagamit sila ng sterilization in order to kill Clostridium botulinum. Then next, disinfection. So, pag disinfection, this is a destruction of vegetative or non-spore forming pathogens, mostly chemically using disinfectant sprays. So, later on, we'll be talking about different disinfectants. Okay? So, the difference between sterilization is that to disinfection, sterilization is killing all microbial life, while disinfection is destructions of vegetative, specifically the non-spore forming pathogens, mostly by the use of chemicals. Okay? Next is antisepsis, treatment of living tissues to kill pathogens or antiseptics. So yun yung ginagamit pag meron tayong mga sugat by the use of other chemicals, just like the betadine, yon, antisepsis or treatment of living tissues to kill pathogens, no, or antiseptic. Control of microbial growth. So we have also another terms, degerming. What is the meaning for degerming? This is a removal of most microbes by swabbing with alcohol. So please take note, degerming, this is a removal of most microbes by swabbing with alcohol. Next is sanitization. So sanitization, this is a removal of most microbes from the surfaces, just like your dishes, glasses, and many more. Okay? So this is sanitization or sanitize. Diba we have a hand sanitizer. So this is also a sanitization. Biocides. Okay? Biocide, this is an agent that kills microbes. Okay? And bacteriostatic, it inhibits microbial growth. Okay? When we call biocides, it kills microbes. But when we call bacteriostatic, it inhibits microbial growth. Yeah. Okay? So, let's move on to our next topic. Okay? The microbial control. So, how to control microorganism? Paano ba natin makokontrol ang microorganism or mga bakterya para hindi natin, no, para hindi ma-spread or para maiwasan natin magdami. Okay? So there are two basic ways. Number 1, by killing microorganisms. And number 2, by inhibiting the growth of microorganisms. Okay? So, cidal agents, which means kill cells. Okay? Bactericidal refers to killing bacteria. Static agents, so inhibits the growth of cells without killing them. Okay? Static. So, ini-inhibit lang niya ang pag-growth. Okay? So, mayroong microorganism but hindi sila dumadami. It inhibits lang ang growth. But the bacteriostatic refers to inhibiting the growth of bacterial cells. Okay? Sa static class, no? Static inhibits the growth of cells without killing them. While bacteriostatic refers to inhibiting the growth of bacterial cells. So, hindi na niya sa bacteriostatic, it's static. So, meaning hindi na nag-grow ang bacterial cell. So, it inhibits the growth of your bacterial cells. Yan. Okay? So, another terms. Okay? Let's talk about sterilization. Mostly, in the laboratory, we are using sterilization in order for us to kill microbial life. Okay? So, sterilization, this is a process whereby all forms of microbial life, including bacterial spores, are killed. Yun. So, that's the difference between antiseptics. Kasi class, sterilization, so we will be killing all microorganisms. It includes spores. Unlike antisepsis or disinfectant or disinfection. So, hindi talaga class sinasali ang spores kasi hindi kayang patayin ng microbial agents ang mga bacteria with spores. Okay? There are different methods in controlling microbial control. So, we have number one, the physical methods of sterilization. So, let's talk about the moist heat. So, moist heat class, when we talk about moist heat, this is a steam under pressure. This is used to sterilize biohazardous trash and heat-stable objects. An autoclave is used for this purpose. Please take note that in moist heat, we are using an autoclave for sterilization. Next is filtration of air. This is accomplished by using high-efficiency particulate air or HEPA filters designed to remove organisms larger than 0.3 micrometers from isolation rooms, operating rooms, and biosafety cabinets. So this is used to control or to sterilize the air. Okay? So we are using HEPA filter or high-efficiency particulate air. Okay? Because it contains 0.3 micrometers of this is they use 0.3 micrometers of filters for the isolation of certain microorganisms. Okay? Next is we are using ionizing radiation. This is used for sterilizing disposables such as plastic syringe or gloves before use. Okay? So there are physical methods for sterilization. We have the moist heat, filtration of air, and ionizing radiation. Next is chemical agents of sterilization. So we have a physical, and now we will be talking about the chemical agents of sterilization. So the chemical agents of sterilization using ethylene oxide. This is used in a gaseous form of sterilizing heat-sensitive objects. Next is your formaldehyde vapor and vapor phase hydrogen peroxide. This is an oxidizing agent. This is used to sterilize HEPA filters in biosafety cabinets. And next is glutaraldehyde, sporocidals in 3 to 10 hours. This is used for medical equipment because it does not corrode lenses, metals, or rubber. Next is peracetic. This is used for surface sterilizations of surgical instruments. Okay? Let's review. We have the physical and chemical agents. So, under your chemical agents, we are using ethylene oxide, formaldehyde vapor, and vapor phase hydrogen peroxide, an oxidizing agent. Glutaraldehyde, this is the best, no, because it does not corrode lenses. And peracetic, this is used for surface sterilization and for surgical instruments. So, these are the chemical agents used for sterilization. Next, physical sterilization. Okay, so number one is for heat. Okay, the function or the purpose of heat is for the destruction of proteins. Moist heat under pressure by the use of autoclave. This is the most reliable. Okay, autoclave. So 121 degrees Celsius at 15 psi for 15 minutes. So the principle is steam under pressure. So question. Okay, question. What tape, no? With biological indicators. So we are using a tape, okay? Autoclave tape with a biological indicator. Please write your answer in our comment section kung ano ba ang biological indicator na ginagamit natin, no? As an indicator that our autoclave is working well. Okay? Please write your correct answer under our comment section. Okay? Pakisulat, no? Pakisulat kung ano ba ang biological indicator. Kasi pag sinabing indicator, ito yung ginagamit natin in order for us to make sure that our autoclave is working well. Kung naabot na ba ang 121 degrees Celsius. Sige. So, I will be giving you enough time to write, okay? To write the answer in our comment sections. Okay? Yun. Okay. So, thank you so much. The correct answer is Bacillus stearothermophilus. Okay? Or Bacillus stearothermophilus. This is a biological indicator na ginagamit sa ating autoclave tape. So, as you can observe it here in our picture, as you can observe it here, okay? So, you can see my mouse pad, okay? So, before, there is no black coloration. But after, you can observe that the line, okay, the line faded, no, ang line na faded color gives a color black, okay? It indicates that it reaches 121 degrees Celsius, meaning that our autoclave is working well. Okay, please take note of this. Right. Yan. This is a typical example of an autoclave. Okay. Next is sterilization. So, fractional discontinuous intermittent sterilization with the use of Arnold's sterilizer. So, it gives us a 100 degrees Celsius per 30 minutes for 3 consecutive days. Ang tagal, class. Because it uses an Arnold sterilizer for almost 3 consecutive days, no? 100 degrees Celsius per 30 minutes for 3 consecutive days. So, this is a sterilization. So, day 1, it destroys vegetative cells. Day 2, it destroys the spores. And day 3, it destroys the remaining spores. So, this is what we call sterilization. Okay? So, its function, no, is to sterilize with the use of Arnold sterilizers. 3 consecutive days natin tong gawin. Dahil in day 1, it destroys vegetative cells. Day 2, destroys spores. Day 3, destroys remaining spores. Under your physical sterilization, we have also an inspisation. So, it destroys cell membrane with high protein content, 75 to 80 degrees Celsius for 2 hours in 3 consecutive days. So, the principle is thickening through evaporation. This is the principle of inspisation. So, under your physical sterilization, we have also the dry heat. Okay? So, we have the direct flame, loops, or needle. Ito yung ginagamit. Okay? Ito yung ginagamit sa mga laboratory if we are inoculating, okay, microorganisms, the loops or needle. We have also the oven. This is used for glasswares with a degree of 160 to 180 degrees Celsius for 1/2 to 1 to 2 hours. So this is the time that we will be using for disinfection or for the sterilization rather of your glasswares. We will be using oven, okay, with a degree of 160 to 180 degrees Celsius. Next, we have also an incineration. So this is used for infectious waste, burning materials into ashes with a degrees of 870 to 980 degrees Celsius. This is an incineration. Okay? So another method, please take note, dry heat method, using the direct flame, using the loops and needles. Oven, this is used for glasswares, sterilization of glasswares, and of course, the last but not the least, incineration. This is used for infectious waste. Sterilizing oven, this class 100 sterilization oven is used for the deep pyroregenations of glass, Teflon, and other heat-resistant materials. So this is an example of a sterilizing oven. This is used in a big hospital. So under your physical sterilization, we have also an ionizing radiation. This uses a short wavelength, high energy gamma rays used to sterilize plastic syringes, gloves, and catheters. Next is filtration, body fluids, vaccines, and radioisotopes. So we have a liquid solution, the cellulose acetate or nitrate medium with vacuum, and of course, the air HEPA filter that removes organism with a size of greater than 0.3 micrometer. Next, we have also a liquid filtration. When we have a solid or filtration, we have also a liquid filtration by the use of culture media, laboratory reagents, and pharmaceutical products. Let's talk about gas filtration. When we talk about gas filtration, we are using the HEPA filter or the high efficiency particulate air filters. And these are used in a laminar flow hoods as well as a hospital room, especially in a isolation room. Most typically, right now, very good example for those who are positive with COVID-19 because the room of an isolation room of a patient suffering from that diseases are filled with HEPA filter in order to filter the air that has been circulated in that specific room. So they are using the HEPA filter. Next, we have the chemical sterilization. So we have an ethylene oxide, formaldehyde vapor, cleansing of, and of course, vapor phase, water, HEPA filter, and glutaraldehyde paracetic acid. Yun. So those are a chemical used in a chemical sterilization. Next, let's talk about disinfection. Okay? A while ago, we talked about sterilization. So, sterilization, it kills 100%. It kills in microorganism, including the spores. Okay, let's talk about disinfection. Disinfection, this is a process whereby pathogenic organisms, but not necessarily all microorganisms or spores, are destroyed. As you can observe it here, we have an example here, non-disinfection agent. We have a Lysol. This is the most common use disinfection agent or the isopropyl alcohol. If nagtitipid tayo, we can use naman the Zonrox, no? Basic example, the sodium hypochlorite. So 1:10. Okay? So the ratio of 1:10 or 10% of sodium hypochlorite solution. Yun. So kung walang Lysol, we can use sodium hypochlorite solution or Zonrox. Okay? So this is a very good agent for disinfection. But always take note, when disinfection happens, there is no destruction of spores. Okay? So unlike sterilization. So physical method of disinfections, boiling at 100 degrees Celsius for 15 minutes, which kills vegetative bacteria. We have also a pasteurizing at 63 degrees Celsius for 30 minutes or 73 degrees Celsius for 15 seconds, which kills food pathogens. And using non-ionizing radiation such as UV light or ultraviolet lights. So, in chemical methods, we have alcohols, aldehydes, and halogens. Disinfection. This is a destruction of pathogens only. Methods, we have physical method, chemical methods. In physical methods of disinfection, we are using boiling. So we will be boiling that certain glasswares or materials for 15 minutes in 100 degrees Celsius. It will destroy vegetative cells only. While pasteurization, it destroys food pathogens in milk or dairy products without altering the taste. Yun yung ginagawa sa ating mga milk or sa mga dairy products. Under your pasteurization, there are different methods. We have a batch method which boils for 63 degrees Celsius for 30 minutes, and we have also the flash method 70 degrees Celsius for 15 seconds. Ang bilis-bilis nitong flash method. Okay? So we have also the physical method of disinfection. Another example is the non-ionizing radiation. It uses a long wavelength, low energy ultraviolet rays. So example is your mercury lamps. These are commonly used in rooms, especially in the hospitals. Okay? So after the discharge of a certain patient in a hospital, they are using UV lamp to disinfect that certain rooms. Okay? So the chemical methods of disinfection. We have number 1, the alcohol. We are using 70% ethyl or isopropyl. So it needs water to destroy proteins. Okay? So we will be using a 70% concentrations of ethyl or isopropyl alcohol because water will destroy protein. Okay? We will not using at 100% alcohol because it will not destroy microorganisms. We need to dilute it in a certain water. But as of the moment, we have a commercially prepared 70% ethyl or isopropyl alcohol. Next is halogens. So example is iodine or betadine. So chlorine or sodium hypochlorite solution, just like your zone rocks or 0.5% or 1:10 dilution for laboratory surfaces. Ito yung ginagamit ng mga medical technologists or even nurses sa kanila mga stations if they disinfect their area. Pag walang mga alcohols, we can use alternative. Okay? We can use sodium hypochlorite solution or your 0.5% sodium hypochlorite solution for your laboratory surfaces. Prior to water analysis, neutralizes with sodium thiosulfate. So alcohols, 70% ethanol or isopropanol or rubbing alcohol, the mode of action is for protein denaturation and coagulation, therefore membrane disruption. Therefore, the microorganism membranes or the membrane of a certain microorganism will be disrupted because the function of your alcohol is for protein denaturation and coagulation. So the advantage of this is for easily evaporate. Okay? After disinfection, as you can observe, when you add alcohol to your fingers or to your tables, it can easily evaporated. So poor effects on wounds, superficial coagulations of proteins. Therefore, it secludes deeper layers of microbes. So it is your alcohol has a poor effects on your wounds because there will be a superficial coagulation of proteins. So the inner bacteria or the inner microorganisms will not be included, right? So it will not be included, but it secluded the deeper layers of microbes because the the first layer of your skin, which contains a lot of microorganisms, alcohols take effect immediately because of a superficial coagulation. Next is salts of heavy metals. They use copper algicides, merthiolate or mercury, silver, 1% silver nitrate or Krebs prophylaxis. It prevents ophthalmia neonatorium. Okay? For your Neisseria gonorrhea. Next, we have the quats or the quaternary ammonium compounds. So, or the benzalkonium chloride. So, this is a non-sporicidal and non-tuberculoidal. So, these are some examples of chemical methods of your disinfectants. And of course, we have also a phenols, the standard disinfectant for your Staphylococcus typhi. The phenol coefficient, this is an expression of bactericidal properties of disinfectants composed of pure phenol. It has the highest dilution of 0.1%. It kills organism at a given period of time. So the phenol coefficient with the highest dilution of 0.1%. The highest dilution of phenol kills organism at a given period of time. Greater than 1.0, better than phenol. Less than 1.0, phenol is better. Yun. Okay? So, equals to 1.0, same efficiency. So, the best example of this is phenol. Okay? Phenol agent. This is a standard disinfectant for your Staphylococcus typhi. How does it work? The efficacy evaluated via standard test method, as we called as a standard organism. Phenols and phenolics, the mode of action is it damages lipid cell membranes. Please take note, the function of your phenol and phenolics, it damages your lipid cell membranes. It's quite effective in the presence of organic materials just like pus, saliva, and feces. So, it is better to use when there are organic materials just like pus, saliva, and feces. Example of this is your Lysol or the orthophenylphenol, or this is a very good surface disinfectant. Okay, so the very good example of your phenol and phenolics is your Lysol or the orthophenylphenol. This is a very good surface disinfectant. Next is your soaps. Your soaps, okay, so the most common soaps advertised by medical technologies is your Safeguard. Soaps, the function of your soaps is for a mechanical removal of microbes through scrubbing. Okay? So soap breaks oily film on skin or as what we call emulsification. Little value as an antiseptic. Detergents more effective against gram positive than gram negative. Always take note that your detergents are more effective against gram positive than your gram negative microorganism. Non-ionic detergents have no germicidal effects or activity, while the anionic or acid sanitizes food and dairy industries. The cationic or positive, okay, charges, this is are used for antiseptic for skin, instruments, and utensils. Okay, so that's the end of our presentation for our bacterial control. See you to our next discussion. Bye-bye!